All right. So good evening, everyone, or good morning, and welcome back, or welcome for the first time to this year's virtual Podium Asia Pacific. I'm Anne Ben from SLING, the Singapore Lipidomics Incubator, and it's my great pleasure to start introducing our first keynote speaker tonight. But before we do this, I'd like to give a big shout out. Tusi, if you could go back to the first slide. I'd like to give a big shout out to the organizing committee, among especially for Tusi. Tusi, super multitasker, pulled all of this together. Thank you very much, Tusi. And we will have the format of this year's virtual podium is on the next slide. So we will have a keynote presentation, followed directly by the Q&A session with Erin, followed by 4 lightning talks with 1 Q&A session for all lightning talks together, followed by the second keynote presentation, followed by another Q&A session and the award ceremony. So with that, it's my great pleasure to introduce Associate Professor Erin Baker, and Erin is a total superstar. Erin is you're Associate Professor at North Carolina State University, and scientifically, Erin has been really successful in driving the field of ion mobility mass spectrometry, having published over 140 papers in this field and also having received 7 US patents. She's also super active in community building. Currently, she serves as the Vice President of Education for the International Lipidomics Society and also as the Chair of the Events Committee of the Females of Mass Spec. And of course, she also serves on the board of various important journals, and very well deserved. Erin was included in the Analytical Scientist Top 100 Power List in 2019 and again in 2021. Erin's research group at North Carolina State University is basically combining advanced separation by mobility, multi-omics analysis, and also software tools to study connections between human health and the environment. And with this, Erin, over to you.
Oh, thanks for the great introduction. And let me pop on.
I'll stop sharing so you can share your screen.
Awesome, there we go. Oh, thanks so much for the great introduction, Anne. And it's such a pleasure to be with you guys all this morning. It's nice and early here, but I know it's really late in other places. So thank you so many people for tuning in. I get to show some really cool research we're doing right now on stem cells. And so I was teasing Anne that I didn't use ion mobility in the title, but we use it in the talk. So you guys will see it pop up here. But before I get going, I have to give a huge shout out to my group. They're the ones that do all the hard work so that I actually get to come and talk to you guys and showcase their cool stuff. So we're going to talk about some ion mobility today, some software, and then some actually biomarker or lipidomic changes as stem cells differentiate. So like Anne said, my group likes to understand molecules present and changing in biological and environmental systems. And that means we do evaluations all the way from exposomics, so things you bring into your body that you might not even think about, pesticides that are on the lettuce in your salads, the air you breathe or the water you drink, and all kinds of other molecules that we might not even think about, all the way down to our metabolomic changes. And so some of the questions my group is really interested in, though, are relating these human health type of questions to understanding these molecular changes. And so some of those questions are, why is a patient sick? How should the patient be treated? And can we detect the diseases before a person becomes symptomatic? And we go through these 5 different steps every time we do a different experiment. As I make my students sit down for sometimes months to do their experimental design. So we always think garbage in is garbage out. So we have to be really smart in every one of these steps. Then we do our data collection, whether we're going to do ion mobility or liquid chromatography, mass spectrometry, then go into our data analysis, our data interpretation, and then finally our conclusions of what we're finding for that study. And so when we're thinking about biomarkers, and this word is somewhat controversial in some cases where people don't really want to talk about biomarkers anymore, but we think it's important to think about these molecules that are changing in control and disease patients, and usually biomarker panels where we're not just looking for one specific biomarker, but maybe 12 to 20 different ones that are changing in the different groups, so that we can actually get a better understanding of patient perturbations. And so that's what we're going to talk about a little bit here too. So thinking of this, we started wanting to do some work with stem cells, and so especially pluripotent stem cells, because they can differentiate into 220 unique cell types in humans. So you can have them differentiate into your neurons, which we're going to talk about today, or other types such as your liver cells, placental cells, and understand diseases by looking at what the normal is for differentiation, and then do alterations to see how those stem cells change. So there's 4 main different areas for stem cell differentiation. You can use that, or for stem cell studies, you can use them in drug targeting, in therapeutics, biomarker identification, and then also disease modeling. So we're going to combine the disease modeling and the biomarker identification. And when we think about neurodegenerative diseases and why we would want to see how a stem cell changes into a neuron, we can really think about Alzheimer's disease. And Alzheimer's disease and many of these other neurodegenerative diseases, such as Parkinson's, have been identified, but cures continue to remain elusive. So we want to use these stem cells to understand what's normal, and then use our analytical techniques to really probe into those molecular changes that occur if we alter our stem cells through their differentiation processes. And so in this, for our pluripotent study with lipidomics, we're really interested in looking at these lipid changes because the brain is very lipid rich. So usually it's thought that over 60% of your brain is lipids. There's also lipids involved with trafficking, so molecular signaling. And there's been many that have been also implicated in these neurodegenerative diseases. Even though we think about Alzheimer's, a lot of times we think about proteins such as A beta or tau, there's also lipids that are also implicated. So when we look at pluripotent cells, here's a nice little fluorescent picture. We can see how cute they are, kind of little round cells. But we know changes have to be occurring when they go to neurons or neural cells, because this nice little fluorescent image goes more to this type of picture. So there has to be a lot of restructuring that's occurring in order to get those pluripotent cells to the neural cells. And so in this longitudinal study, as we followed it through, what we did is we took different time points through through the stem cell development. So we started with day 0 with pluripotent stem cells. Then we went to day 3 and day 7 and took samples at both of those. And this is when those pluripotent stem cells change into embryonic bodies, or we'll call these EBSs. And this is a very important changing time, because then they're just started, you can really alter them very easily to change into things like those liver cells or kidney cells. And we're just forcing them in certain ways in order to go to these neural cells. And we were working with Kevin Francis in this study from Sanford Health in South Dakota. So then we also followed them into the day 21, which is more the neural stem cells, and then finally to day 42, where they turned into neurons. And so for those of you that aren't as familiar with lipids, because I know this is an omics type of talk, I wanted to talk a little bit about why we are interested in lipids. And the way that we can all kind of focus ourselves is we think about lipids when we go to the doctor and do our yearly exam, and they start looking at your triglyceride level or your total cholesterol level, maybe your LDL or your HDL. And this already shows you that they know lipids are really important for your health. But what we really want to do is we want to dive into which specific lipids are good for your health. And we know that these lipids can change based on the foods that we eat and the diet that we have. We also know that things, just as cell signaling or cell replication can change the lipids that you have in your body. And so if we go to lipid maps, there's 8 different categories of lipids. So it's not just your LDL, your HDL, or your triglycerides. There's many different types. So two different ones here are the glycerophospholipids and sterile lipids. And here's our cute little signaling lipid that's sending messages back and forth into our cellular membrane. Then if we go specifically into one class of lipid or one of those categories, so if we pop into the glycerophospholipids, we can see that that class has many ways to vary itself by its head group, where you can have PCs or phosphatidylcholines, PGs, phosphatidylglycerols, and then many, many other different head groups onto those glycerophospholipids. And then finally, we can change the fatty acyls on those lipids. And so now we have different numbers of carbons and different number of double bonds. And so you guys will see commonly this nomenclature of 22,6 or something similar, where the first number is the number of carbons in your fatty acyl, and the second number is the number of double bonds. And this is the common fatty acyl nomenclature with lipids. So now what we want to do is we want to look at all these different possible species of lipids just using multidimensional separations. And so this is where the ion mobility comes in, because we love having a structural component also in our measurements. And we do all of our evaluations with a C18 reverse phase LC on the front end to give us really good polarity separations. Then we add in the ion mobility to do size separations. And then we do CID, TOF MS to do fragmentation and MS evaluations. And so for those that aren't as familiar with ion mobility, we use drift tube based ion mobility. And what that is it has a drift tube, of course, and it's a little bit opposite of mass spec, where in mass spec, you try to pump out all the gases in the drift tube. In drift tube ion mobility, we actually try to put as much gas as we possibly can in until we start losing sensitivity. So we like to do a low pressure version of ion mobility that's about at 4 torr. And then we apply a weak electric field so that the molecules can actually be pulled across to the end. And you have to do this in basically a race. So you have to have a start time and an end time. So we inject a square wave pulse with our molecules in it, and then they'll be pulled. And here we have 2 different fatty acyls, one with a trans double bond, one with a cis double bond, and we can see they have 2 different orientations. So when we inject them into the drift tube, our smaller one with the cis double bond is pulled through faster than that with the trans double bond. And we can see shape separation instead of just the M over Z, which would say from the mass spec would just say that we have that specific M over Z present. So we get this nice value called the collision cross section. And that allows us to compare our measurements with everybody in the world, basically, because we've been able to standardize based on the pressure and temperature we used and the length of our drift tube. However, these measurements can be really challenging as we can get really large file sizes. And so it can be really manual to look into those. So what we've done is we've built a really nice database with Skyline, and we just had this paper accepted in JPR last week with our lipid library so that people can actually use this, and it's going to be publicly, or it is publicly available. But we have about 954 features. We actually might even have a few more now. This might not be the latest number. And of those, 616 are unique lipids. And why I say 954 features is you see some of them in positive mode and some of them in negative mode as different ion types, but it goes down to 616 different unique lipids. And in our Skyline library, each one of these has its different MS-MS transitions to it, a collision cross section, and then also the mass spec information. So you'll be able to search each one by the different dimensions, and then also the LC retention time. And you can see all the different lipids that we've annotated here, and then how they look as far as collision cross section and M over Z spread in both positive and negative mode. So when we get back to our analysis of our stem cells, though, what we were able to do from this library of close to 1,000 close to 1,000 features is we were able to annotate 350 unique lipids and their abundances in each one of those steps, and not each, but I guess total from the different steps. And then we wanted to compare and see how the changes were occurring between the different steps as the stem cells were differentiating. And so if we did just a quick plot, a PCA plot, to see if we saw differentiation based on lipidomics, it was actually pretty cool because we did. So if you look at our pluripotent, that's our nice little pink one here, then we went to the different embroid bodies, and they're over here in our PCA, then our neural, and then finally our neurons. So we know that the lipids are actually differentiating the different steps in differentiation. So now we want to understand which species are actually pulling these guys apart. And so then we pop into another tool that we've developed to help us visualize lipid changes, which we call structural based connectivity and omic phenotype evaluations. And this is in a paper in Analyst, but also is available through GitHub if anybody likes to code. We're working on a web-based version of this though, so people that don't like to code can actually use it too. So what SCOPE does though, is it tries to understand the differences between head groups and fatty acyls and if there's a common connection between lipids that are changing. And so we'll look at like this specific phosphatidylcholine that has an 18,0 fatty acyl and an 18,1 fatty acyl and see if it varies or changes differently than maybe a different phosphatidylcholine or maybe a triacylglyceride. And so then can we take these head group information and this fatty acyl information and pull out important biomarker or dysregulation observations? So how SCOPE works, and the part that I'm going to show you today, is it uses hierarchical clustering. And in this clustering, we're putting things that are similar together so that we can visualize them in a very nice and easy way. And so in this hierarchical clustering, if you had 6 different letters, we first want to look at the distance between each of those letters. And how we're going to do that is through smiles for structural differences. But here, just to simply show it, we're just going to look at the length between these. So we'll first group E and F because they're the closest together. So they're going to be the lowest branch on our dendrogram tree. Then we're going to group A and B. They're the next closest. Then D to E and F. And C to the D, E, and F, and then finally group everything to the final dendrogram. And how this will work with our lipids is basically a lot of the clustering is going to be based on the head group because the head group is very different in atoms than our fatty acyls. So the clustering is first going to look at these 4 different lipids, and it's going to say these phosphatidylcholines are the closest in structural relationships. Then it'll go to the phosphatidylethanolamine, and then finally the ceramide, and you get everything grouped into this dendrogram. And this has really helped us with visualization because it's really hard looking at huge Excel lists and then looking at p-values and log fold changes. So finally, this resulting dendrogram is basically driven by the head group of these lipids. And so I'll show you one of those really quickly, but first, when we started looking at the statistically significant lipids, what you'll do before you do SCOPE is you do all your identifications and then you do your statistical significance and then you bring those into SCOPE to actually visualize those. So when we looked at our different longitudinal steps to go to our neuron, we found 193 statistically significant lipids, not all in each step, but throughout the whole longitudinal time range. And then we did our dendrogram. And so for these 193, you can see that our dendrogram wraps them in a nice little wheel. And then it's doing this clustering based on head group, where we're seeing our fatty acyls grouped, our ceramides, our hexceramides, our sphingomyelins, and then as we go down to our triacylglycerides. And you're going to say, okay, that's kind of cool. You know, we've grouped our statistically significant lipids, but what does that mean? So what we say that SCOPE is actually doing is it's going to combine basically a volcano plot, and then it's also going to combine a heat map to show you what's going on. So when we take our statistically significant changes between our pluripotent stem cell or lipids and then our embroid bodies at day 3, what we do is we add a rung to this, and we see that 50 are going up and 44 are going down, and then we add our rung to our dendrogram. And so we see all of the other ones that were identified, and these are going to be in the different steps that aren't statistically significant are shown as gray. Everything that's up is shown as red, and everything that's down is shown as blue. So we can easily look at this and say that our plasmalogens are showing a lot of upregulation in this change from pluripotent to the embroid bodies. Where if we go to our phosphatidylcholines, everything that's statistically significant is showing as blue or downregulated. Now we can keep adding rungs though, so we can do comparisons across the different stages. So then if we go from day 7 to day 3, we also see changes in these plasmalogens. But now we see one of our phosphatidylcholines going up and not very much dysregulation in there. But if we look at our outside, we see a huge amount of dysregulation in our triacylglycerides. Now, when we go, we saw a huge change though when our stem cells went from day 7 to this initial neural stem cell. And we can see that our triacylglycerides completely reversed what they were doing in our first step from day 3, our pluripotent to day 3. So it was really interesting. And then also our PEs, which were downregulated in our first step, completely changed to everyone being upregulated in that change to the neural stem cells. So this has really shown us the difference between the different steps and the different time points as we do our longitudinal analysis. So are these lipid changes unique to neurons? That was another question we got. Is this just stem cell differentiation, or is this actually stem cell differentiation to neurons? So we went to mesodermal cells, and we looked, and we also see differences in the PCA plots for the mesodermal cells, which are the muscle cells if you're not used to cell terminology. And as we looked into these, I'm not going to show it just because time is short today, but we are seeing completely different changes throughout the different steps as we do longitudinal analyses of the mesodermal cells. So we're really excited that by using this visualization tool and then actually just looking at the lipid changes that are occurring between the different stages in the stem cell progression, we're seeing huge differences on how these lipids are reformatting. And so just to give you some conclusions, so the lipidomics helps us understand the unique mechanisms for the neural stem cell differentiation. And now we're following other stem cell lines and other stem cell types. And it's helping us define what's normal for a neural stem cell. So now we're going to also start altering some of our neural stem cell steps as they're longitudinally growing. So we can see if we can understand what's actually going on molecularly in the different processes. And so finally, a huge acknowledgement to my team, all the people that do this and the funding that we have. And a big thanks to everybody that stayed up or woke up early to listen to this. And I'll turn it back to Tusi and be happy to answer any questions.
Thanks, Erin. That's great. While we are waiting for other questions, Erin, can I ask you questions?
Yeah, definitely.
You mentioned that tags are changing. Is there any connection to DAGs? Is there a relationship between tags and DAGs changes, or?
Yeah, good question. So we thought that too. We're like, the triacylglycerides are changing so much in the step 1 and step 3 that I showed. And so we would expect some DAG changes, but there are very few DAG changes. It's mainly in the tags. So we're looking into that further, but it really is mainly showing up in the tags right now. We do have more tags in our database than DAGs, and so we're trying to populate that a little bit more, but we aren't seeing as many DAG changes.
That's great. We got questions from Krishante. Thanks for the nice talk, Erin. I have two-part questions. You see isomeric separation on the chromatograph scale.
Let's see.
That's all. Okay. Second, any isomers identified as biomarkers?
Gotcha. Yeah, and so we do see isomeric separation, especially in the lysolipids. So we can see the SN1 and the SN2 position changes in the LC. So that we like that because that pulls them apart. We also see a tiny bit. We do 35-minute LC analyses, so we see a small amount in double bond position, where usually if you do the XIC, you'll see kind of a big fat peak, and usually at the beginning we'll see one double bond, and at the end of the peak we'll see another double bond, which is also slightly validated with our ion mobility isn't the highest resolving power, but we do see a slight shift in those peaks with the ion mobility. And there's, I'm sure with the new versions from Mobile Ion, and then we're also using the high res demultiplexing with the Agilent, we're able to actually get better separation there too. Right now, we have isomeric biomarkers in other systems, but we don't have it in the stem cell differentiation yet. But this is pretty fresh. So we've only been these results we probably only had available for maybe a week and a half, 2 weeks. So we are looking in deeper. A lot of the isomeric validation takes us a little while because we really have to manually dig into it. So yeah, really great questions.
Thanks, Erin. Yus here, Yus asking, Erin, how do you control quantitation?
Yeah, that's another really good question. And so we do some spike ins. So with the different splash mixes, so we really like the Avanti new version that has the 64 heavy label. That one's really good, but it's not possible in all the measurements we do. And so when we can't do that, what we do is basically a TIC normalization is the best type of way if we can't do the spike ins, or we'll spike in just a few things and do the quantitation, but there are times where it's just not possible to do the spike ins. We wish it was possible every time, and I know that's most people would say they do it every time, but there's times where our spike ins cause a few problems with our analyses.
Yeah. Any questions from please type or while I'm waiting, because we got 2 minutes, so I'm happy to ask more questions. So James, you want to ask question?
Yeah. So did you say use the smiles to do the hierarchical clustering for the structures?
Yep.
How do you read a smile? And I'm just like, I don't get this. I'm never going to understand. Can a human read a smile, or is that really something you got to put into a piece of software to interpret?
Yeah, that's a great question. So my students that do this all the time can actually read them. I can't
Okay.
either. I look at them and they just look like a bunch of equal signs and C's and like all kinds of crazy letters. But my students that have done them can actually read them. And the hardest thing was there were a bunch of lipids that didn't have smiles. So we had to go through ChemDraw and make smiles for them too. So when we initially started, we were only able to do dendrograms for a portion of those. And so that's why if anybody wants to do this, please get our information from GitHub, because they did probably 2 to 3 months of work to get smiles for all the lipids in order to be able to put them into dendrograms. Because yeah, we would have people just drawing everything up in ChemDraw to make sure that we could get all of them out.
Yeah, it's a huge amount of work, but that, like you said, that visualization is so powerful. I could see everything that you were saying straight away. Yeah, really beautiful, really nice.
Yeah, we were having such a hard time when we first started, because you just had Excel sheets, and then the more patients you had, the more tabs basically, or rows or columns and things, and it was really, really difficult. But yeah, we're trying to work a lot on this chem informatics visualization, because especially when you get into multi-omics too, it's too hard for a person to process all the data, and so at least this lets you get the high points.
Yeah, definitely.
Thank you so much, Erin. So it's 11:30 here. So it's time for the lightning talks. Our first lightning talk, apologize for my pronunciation, Chon Lee from Beijing, China. Thank you so much for accepting the last minute. So you can share your screen, Sean. Yeah.
Hey, hello.
We can hear you. Yeah, thank you.
Okay. Hello, everyone. I'm Chao Honglim from Tsinghua University. It's my great honor to share my research on this lightning talk session. I would like to talk about my recent research about deep profiling of aminophospholipase from biological samples while to orthogonal derivatizations. P and PS are glycerophospholipids harboring a primary amino group in their head groups, collectively termed as aminophospholipase. They are key components of cell membranes and play roles in numerous cellular functions. Aberrant APL profile also correlates with cancer. APL are susceptible to ion suppression, and the PS suffers from pigtating on the LC colon. In this work, we developed a novel derivatization strategy for APLs using TMAP NHS reagent. The introduction of amino group enhanced the detection sensitivity and significantly reduced pigtating of PS on the helix colon. Based on this, we established a sensitive and structural informative LC-MS workflow for APL profiling. We integrated offline TMAP derivatization with helix separation and the online acetone Padano-Bucci reaction to obtain cis-adult bond location information from subsequent MS2CID experiments. The relative composition of cis-adult bond location isomers was calculated from the relative abundances of cis-adult bond diagnostic ions. This workflow was applied to the analysis of APLs from prosenbrin lipid extract. For example, based on chain composition and the cis-adult bond location information, PS361 can be identified as a mixture of these three cis-adult bond location isomers. And another example shown here is PE postmologen. As a result, the workflow provides detailed structural information, detailed structural annotation of more than 200 APLs from prosenbrin lipid extract at the level of cis-adult bond locations, including 74 PS, 59 PE, and 87 ether PE. As compared to the results obtained without TMAP derivatization, as shown by the columns labeled with N, TMAP derivatization greatly improves the coverage for PS and ether PE species with medium to low abundance. That is relative intensity less than 10%. We next employed the workflow to map the relative compositions of cis-adult bond location isomers between normal breast cells, HMEC, and breast cancer cells, 231. Our data revealed elevated contribution of N10 isomers of fatty acid 16.1 and 18.1 in cancer cells, which is correlated with an increased transcription of fatty acid desaturase 2, FAST2. FAST2 catalyzes the formation of cis-double bond at N10 in fatty acid 16.0. The elongation of 16.1 N10 forms 18.1 N10. When cancer cells were treated with a small molecule inhibitor of FAST2, the relative compositions of most FAST2 related products were found to be decreased. The data may provide new insights into understanding the role of FAST2 in tumorigenesis of human breast cells. Overall, the presented workflow is capable for in-depth analysis of APLs down to cis-adult bond locations, and it has the potential to linking the missing dose between aberrant APL profiles and the lipid reprogramming in disease. Thank you for listening.
Thank you so much. We will ask questions all together, all the lightning talks. So we will move to the next lightning speaker. Hamsha Hassin Ahmed from University of Florida. Hamsha, take away you can share your screen. Thank you for joining.
You see my screen?
Yeah, presentation.
Okay.
Yeah, great.
Yes. Well, good morning, good afternoon, and good evening for everyone around the world. And I would like to thank the organizing committee for giving me the chance to be part of the presenters today. My name is Hamza, and I work in the Garrett Lab at the University of Florida. And my talk basically is about a small part of my PhD work, which we are evaluating the role of Fusobacterium species in colorectal cancer metabolism cell line and cell line metabolism. So a little background of the gut microbiome and colorectal cancer. And this cartoon represents the path from health to solid tumor malignancies at mucosal sites and the microbiota contribution. Signaling changes in the colon epithelia occurs as a result of pathogen-driven metabolites. The infection can lead to invasion, which contribute to breach of the body's mucosal barrier. And failure to repair this barrier breach will lead to failure of restoring the homeostasis. Therefore, the microbiota can influence tumorigenesis by different method or characteristic, but we are focusing on metabolism, and the microbiota can modulate the metabolism within the host. And recent studies have shown that Fusobacterium species are implicated in the onset, development, and activating metastasis of colorectal cancer. They activate the cell proliferation using the Wnt signaling pathway. They also interact with the host cells to localize on the cells. Also, they can modulate the innate immune system. And once the bacteria get into the cells, it increases autophagy, which lead to chemoresistance. These are the major activities that have been reported regarding the F nucleatum mediated colorectal cancer. Thus, we hypothesize that Fusobacterium species can reprogram the metabolic network of colorectal cancer cells and subsequently promote colorectal cancer tumorigenesis. So we conducted HPLC, ultra high pressure liquid chromatography, HRMS-based metabolomic experiment, and we chose the colorectal cancer cell line CT116 and two species of the Fusobacterium, F. periodontum and vincenti. The 116 cells were infected for 24 hours, which is the time needed for bacteria to reside in the host cells. Samples were collected and protein precipitation was employed to extract the metabolites. Metabolites also were separated using the reverse phase chromatography and positive negative ionization mode, electrospray ionization mode were performed. In the results section, we can see in the score plot here that from the principal component analysis showing the identified feature of positive and negative ion modes, there was a clear separation between the infected and non-infected colorectal cancer cells, which indicate that the infection influenced the metabolome of colorectal cancer cell line. Since we saw this separation, so we conducted and generated a heat map containing the top 50 differential expressed metabolites. So many of the significantly clustered metabolites were found to be amino acids, nucleosides, nucleic bases, amino sugars, polyamines, glycerophospholipids, and carnitine and its derivatives. But most importantly, some of these differentially expressed metabolites related to energy, such as glutamine, since glutamine is a precursor of glutamic acid, possible explanation for this might be that Fusobacterium species catabolize the glutamine to glutamic acid and provide energy source for the cells and for the bacteria. Finally, we were able to generate a metabolic pathway analysis, and you can see these figures illustrate the summary of the top dysregulated metabolic pathways pathways that in the colorectal cancer cell line caused by the Fusobacterium species infection. To summarize, these findings suggest that specific species of Fusobacterium in one cancerous colon site can alter the colorectal cancer metabolism. And I just want to thank, this is my acknowledgement, and I want to thank my lab members in the Garrett Lab, and special thanks to Sasanka and Laurel Mack and everyone. And that's all. Thank you for listening.
Thank you so much. We're getting questions, so we will get back to those questions soon. So our next lightning speaker is Gina Barbosa. She's from Central Mindanao University, Philippines. Gina, over to you. You can share the screen.
Good evening, good morning to everyone. It is an honor to present to this distinguished, can you hear me? Sorry. It's an honor to present this to a distinguished audience. At this onset, I would like to say thank you to Thusi and to the rest of the organizing committee for giving me this opportunity. From common to rare gingibraceae plants, a metabolomic study using GCMS. Gingibraceae plants, commonly known as gingers, are very popular because of its culinary and medicinal applications. However, there are still many ginger plants, especially the rare and Philippine endemic gingers, which are still left scientifically unstudied. So this study aimed to investigate the metabolic composition of and determine the differences between leaves and rhizomes of a range of gingibraceae plants. For the method, untargeted metabolite profiling by gas chromatography, mass spectrometry was conducted, and the statistical analysis of the data generated was done using metaboanalysts. The plants used for the common gingers, Gingiber officinale, Curcuma longa, Etlingera elature, and for the rare species, we have the Philippine endemic Hornstigia conidea, Etlingera philippinensis, and the rare Amomoricarpum. The results of the study, as shown here in the heat maps generated using the hierarchical clustering analysis, it is shown that the Etlingera elature and Etlingera philippinensis belong to the same subclass. This supports the fact that these two belongs under the same genus. We have the two common Curcuma longa and Gingiber officinale also belong to the same subclass. For the rhizomes, it can be shown here that the common gingers, Gingiber, Curcuma, and Etlingera elature fall under the same subclass, indicating close relationship among these ginger species. The categorization of metabolites according to compound classes is shown here. As revealed, we can see that there are actually lots of percentage of unknowns detected both in the leaves and rhizomes. And a total of 130 metabolites were detected both in the leaves and rhizomes of gingers. And we have actually 76 known, and we have 36 unknown in the leaves. We have also 64 known, and we have unknown 22 from the rhizomes. For 44 of these metabolites were uniquely determined or in the leaves, and we have 18 metabolites detected only in rhizomes. As can be seen here in the right side, we have the VIP scores, or the variable importance in projection scores, to determine the chemical markers. The Amomoricarpum leaves display the highest number of chemical markers, and this includes canic acids, canic acid derivatives, and interestingly, there are lots of unknown metabolites. And in this study also, it can be shown that like the transcaffeoyl canic acid, the transchimeral canic acids were actually detected first in ginger species. As can be seen here, we could say that this metabolomic approach is indeed very useful tool in determining the metabolite profile of the under-investigated plants. So the results of the study shows that the significant baseline information are provided that could be helped in assessing the underutilized leaves of the common gingers, as well as in exploring the rare ginger species in the Philippines. In conclusion, this study, being the first metabolomics report on rare plants such as Amomoricarpum, Etlingera philippinensis, and Hornstigia conidea, affirms the usefulness of untargeted metabolic profiling in exploring under-investigated plants. Before I end, let me say thank you very much to Metabolomics Australia, to Professor Utna Rusner, and to the rest of my co-authors. So good evening and good day, everyone.
Thanks, Gina. Thank you so much. So our last speaker for the lightning speakers is Daisy Zahor. Daisy, over to
Hello,
you.
everyone.
You can
Can you
share
hear me?
Yeah, you can share the screen first.
Okay, no problem. Okay, good day, everyone. Thanks for the introduction and invitation. So I'm Daisy Zhao from the Hong Kong Polytechnic University. So today I'm going to share with you some of our recent findings related to the prebiotic effects of phenolic compounds on gut bacteria while using the metabolomics techniques. Okay, so it has been known that the polyphenols from different types of foods, they can have different effects on the bacteria, especially for ellagic acid, which is a commonly consumed polyphenol from foods like pomegranate, berries, and walnuts. They can have wide different types of interactions with gut bacteria. So why we're interested in this polyphenol? Because it is poorly bioavailable. So it needs the biotransformation by gut bacteria to make them more orally bioavailable, and they can exert biological functions like antioxidant, anti-cancer, and anti-diabetes. However, the transformation is limited by the specific species in the gut microbiota. Now, currently, there's only 3 strains that have been characterized responsible for this kind of transformation from ellagic acid to different types of uracils. And the rate-limiting metabolite is the uracil C, which requires the species from Bordetellobacter. So we're interested to know whether there is some interaction between the ellagic acid and gut microbiota. So our previous studies using ellagic tannin-rich raspberry extract shows that the phenolic extract can selectively enrich certain species in the fecal microbiota from different species. So we collected the fecal samples from healthy human adults, infants, mice, and rats. So what we found that after inoculation with the microbiota and the ellagic tannin-rich phenolic extract, certain bacteria, especially like some probiotic strains, including Echinacea, Legionella, bifidobacteria, and lactobacillus, are enriched. However, this kind of beneficial effects have never been explained. Is it due to the biotransformation of the phenolic compounds by certain bacteria that has beneficial effects on these probiotics? Or it is because the bioaccumulation effect that can, for example, store in the bacterial cells to stimulate certain metabolic activities? Or is it caused by the precursor or its microbial tablets? And what are the metabolic pathways involved? So to answer these questions, we use integrated metabolomics approach. For the microbial metabolomics, we try to extract a broad range of human metabolites from the bacterial cells. So here the diagram shows you 2 different types of pretreatment methods. The data presented here is using conventional method because we are still optimizing the new method, which is to use the next generation matrix removal microillusion 96 well plate to remove the excessive amount of lipids, especially those from the bacterial outer layer that can complicate the detection by the high resolution mass spectrometry. So for the traditional methods, we can successfully remove some of the out-membrane lipids, so we can better clarify the small molecule metabolites inside the bacterial cells. So from the one ANOVA analysis, we found like over 300 metabolic features were upregulated. Next, we did the GCA studies to try to cluster different treatment bacteria receiving different treatments. So we can see that for Decornibacter urolithin fissions, which is one of the bacteria that can convert EA into urolithins. They can be all successfully clustered very differently. While for the metabolites, urolithin A and urolithin C, they seem to have a similar effect, which can actually correlate to the bacteria growth effect. And in addition, we also tested the effect on the Echinacea ministophila. So we found that the effect of urolithin C and EA can be successfully distinguished, although there's not complete separation. So what are the metabolic pathways that have been altered? So we found that there's a number of ribonucleotide monophosphate that has been upregulated, which indicates the influence on the cell cycle and cell division related activities. And also, and very interestingly, we found there's a dose-dependent accumulation of phenolic compounds. For example, in G. uro, we found ellagic acid can be accumulated when higher levels of the EA can be treated, indicating that probably the bacteria have to survive in such a high concentration of phenolic compounds in order to utilize these phenolic compounds for certain metabolic activities. While for the Echinacea, we found also like urolithin C, it can be enriched after treatment with the urolithin C. So these all indicate different capacity of the bacteria to utilize different phenolic compounds, and also maybe they can influence their cell cycle, cell division, and amino acid metabolism. And also bioaccumulation of the phenolic compounds were observed as well. So in our future studies, we're going to validate our new method in treating the bacteria samples, and also we hope to extend the application to other sample types of samples. And also we hope to do some lipodomic studies to further understanding the prebiotic effects of the phenolic compounds. Okay, so this is what I want to share with you all. And I'd like to thank all my lab members and our Future Food and Research Institute at the university. Okay, thank you.
Thank you so much. Well, very nice presentation. All the lightning talks, so beautiful. This is the time for Q&A for lightning talks. Please unmute yourself and ask the question. I saw a few questions from you. Do you want to unmute and ask? No? Okay, I'll read the questions then. I saw that questions. Sorry. Erin, can you see some questions? I can't see any.
I saw. I have a question actually. I'm not sure
Yes,
if I
please.
can answer it, but I do have some. I got some questions. So the first question is like, I was asked the lipidomic effects checked with the other cancer cell lines of the same infection. Yes, we did the lipidomic. Lipidomic study for the same infection, and actually the big or the major study was about two different cell lines, SCT116 and HT29, but I just show a piece of that study. And we also did the lipidomic studies, and also we did the helix studies, and we saw, yes, big different effect of the infection on the colorectal cancer in the same way. And I hope that is answering the question for Dr. Adam Maski. Yeah. And I guess I got another question in the chat saying, so I can answer that question is that basically it's a good question. However, like the identification of distinguishing metabolites from the bacteria on the host is kind of still difficult and it's not that easy to do. There is not a specific method. However, we did a flux study to trace it by carbon label. And yes, we found, and I would say, a great results. And we detected a specific metabolite. We could assume, and definitely it's coming from the bacteria. So, and we are starting writing the manuscript for that. And we are on, and we did that actually 3 weeks ago. So it's still a new, fresh data. And we're going to repeat the experiment again for different other metabolites, and soon will be published. I hope I answered all the questions.
And I think there's one more question. How did you discern bacterial metabolite from cancerous cells?
Yeah, that's the last part of it.
Okay.
That's what I'm trying to say. It's like the.
Okay. There's another question from anonymous attendee. This question is addressed to Gina. How would you have furthered your research given that there are unknown metabolites?
Yes, thank you for this question. Actually, this actually is stimulating me because in reality, I was not able to pursue further studies because of our lack of facilities in our place here in Mindanao. But as what I did is I further go other parts of especially the edible parts of these plants for biological activity, and then also I'm looking forward for further collaboration. We have to pursue this more, especially on isolation, because I could see that this unknown metabolites are promising, because in my previous study on its antioxidant, they have very high antioxidant activity, and I'm so sure that these are very interesting compounds. So I'm just looking forward that with our minimal, or we have no enough facilities, I could do further more collaborations to enhance or to maximize the unknown species identification. Thank you for this. That's everyone.
Thanks, Gina. There's another question from you. This is for Dr. Saho. I think that's for Daisy. Have you considered analyzing your microbiome for genetic richness? I mean genetic diversity, species identification, and abundance count of specific bacteria species.
Thanks for the question. It's really a great question. Let me share my screen again, if that's allowed. Okay, so actually, how did we come up with this idea, like why we're going to study the selected bacteria of interest is because we have done the cysteine S RNA sequencing. We found that in fermentation, like in a colon simulator, with the different types of gut bacteria, gut microbiota, can selectively enrich certain genera, a richness in the gut microbiota. So we selected the type strain from these different genera. For example, the LGG from the Lactobacillus genera, and the Giro, because Giro is as I mentioned, is the responsible bacteria for converting EA into uracils. So we are also interested in this bacteria. And then the other one is like Echinacea, which is the Echinacea minisophila. It's a type strain. And lastly, we select the B. infantis for the bifidobacteria as the type strain. So we try to understand how different probiotic bacteria are influenced by lactic acid or its metabolites. So in the future, we are going to do animal studies and further confirm its in vivo effects. I hope I have answered your question.
Thank you so much, Daisy. I think we are coming closer to our second keynote speaker's presentation. So let me share my screen once. It's my great pleasure to introduce Rick Dunn. After seeing a long time, I feel like I'm back to my Metabolomics Society. Rick is Professor of Analytical and Clinical Metabolomics Biochemistry in Liverpool, UK, and he's the chair in Analytical and Clinical Metabolomics at University of Liverpool. And today, Rick is presenting some practical consideration and the next development steps required for high confidence metabolite annotation in non-targeted UPLC MS metabolomics. Over to you, Rick.
Okay, thank you. Thank you, Lucy. Good morning, afternoon, evening to everybody. Thank you very much for the invitation to speak today. I'm just going to quickly rush through three different projects that we're working on here at the University of Liverpool in relation to metabolite annotation identification and thinking about how we can improve our confidence. And really, I'm talking about this from an untargeted metabolomics point of view. Let me just share. Okay, so really I'm going to talk about untargeted metabolomics today, though. Obviously, we should all be aware that untargeted metabolomics is very much a discovery tool that we apply, and then we should go on to validate or further validate our findings within there, or into translation, for example, in clinical practice within there. But I'm going to focus on this today, and I'm going to focus on metabolite annotation within here. And really, you know, in untargeted metabolomics, we're really focusing on trying to measure as many metabolites as we can in a reproducible way, but in a true discovery study, because sometimes there's metabolites which we don't know are important within a biological system, but only through the untargeted can we actually find out. We can't predict which metabolites would be important and therefore develop a targeted assay. Now, there are what we call semi-targeted assays that have been available for a while, things like Metabolon kits, Metabolon systems that they have, Biocotis kits, etc. as well. This is where we know what the targets are, but there's a larger number of metabolites within here. So typically in untargeted, we're measuring low thousands of metabolites, and semi-targeted, we're measuring low to high hundreds of metabolites within here. But if we think about, I'm a mass spectrometrist, so I always use liquid chromatography mass spectrometry or gas chromatography mass spectrometry within here, but I'll focus on liquid chromatography mass spectrometry today. And typically there's three, though we've heard about a fourth from Erin earlier, ion mobility on here, but I'm going to talk about three of the most common types of data that we use. The first one is the retention time. So we use ultra-performance liquid chromatography to separate out metabolites, either partially or fully separate metabolites out over time. And we use the retention time matching to authentic chemical standards. We can use the mass, the accurate mass of the intact metabolite. So that's what I call MS1 within there. And we can fragment the metabolite, the intact metabolite, fragment that into different fragments, which are dependent on the chemical structure of that metabolite, and we can measure that fragmentation mass spectrum as well within there. So I'm going to talk about aspects of all of these as we go through today. And I've said at the start, it's Rick Dunn and friends, and you know, we all perform research as groups or with collaborators, etc. in there. So as I go through, I'm going to introduce who's actually done the work, because a lot of the work I haven't done myself, as you may expect. So the first work is actually mainly from Dr. Will Nash. He performed this during his PhD, and he's doing a postdoc in a group continuing this as well. But also, hopefully soon, Dr. Judith McGeer. She has a Viva in a couple of weeks' time. And I'm going to talk about electrophorate complexity, because sometimes or often this is forgotten, and it can influence the number of false positives or false negatives that we get within our metabolite annotations using just the MS1 data. So I'm just focusing on MS1 data here. So if you took a chromatogram of 10, 15, 20, 30 minutes long and just summed all of the mass spectra across there into a single mass spectrum, this is the type of complexity that we see. It looks relatively complex, and then you home in on a small region of that, it looks even more complex, and you home in on a small region of that, it looks even more complex, etc. And this is because electrospray ionization, it's a bit like an electrochemical reactor. Well, it is an electrochemical reactor, but also what we're introducing into the electrospray ionization source as well. We're not just introducing metabolites, there can be salts, metals, etc. in there, extraction, solution, mobile phase solvents, etc. within there. So there's a whole range of different things, different chemical reactions that can occur within the electrospray ionization source every single second as you're going through your chromatographic run within there. We know that we can detect tens of thousands of features, but that's not tens of thousands of metabolites within there, because one metabolite can generate what we call multiple features. Okay, so one metabolite can be detected at different masses with the same retention time. So the structure of the MZ retention time data can be used to be able to group features together based on the same metabolite within here. Okay. So all of these different features are formed in the electrical ionization source at the same time, and then introduced into the mass spectrometer and detected at the same time. So they should all have the same retention time, and theoretically the same peak profile, though depending on how noisy your data is, that isn't always the case, but they should have the same retention time within there. The second thing is that if you measure these across multiple samples, and the larger the number of samples, the better, and you perform a correlation analysis, a pairwise correlation analysis between different features of the same metabolite, that should be positively correlated. So we can use that to start to group features together as well. And finally, the MZ difference between different features. You can't just have any MZ feature. There's only specific MZ features that you can have. And actually knowing what these MZ differences are between different features is quite important. And I'll come on to that in a minute. But we can use all this information to be able to group features together. So we're grouping all these features into a single metabolite and then reporting for that single metabolite. And it reduces the complexity of the data. So we knew all about this complexity a long time ago. We published on this, blimey, I didn't even have a grey beard then in 2009, but we looked at the early complexity within there. And more recently, Will Nash has looked at this in a lot more detail. He's taken greater than 200 studies. He's looked at grouping all the different features based on the retention time similarity and the correlation analysis within there. And then he's looked at all of the different MZ differences that are available. Okay, or were detected, and he's linked those or sorted those by frequency that are detected within there. And you can start to see there's a lot of different MZ differences that you can detect. There's 547 in Will's data set, and of these, about 322 could be annotated, either as adducts, isotopes, in-source fragments, biological transformations as well, which you'll see in here. If you've got two different metabolites that are biologically linked but have the same retention time, then there's a chance that they'll be positively correlated as well within there. So there's a lot of complexity within there. We're now writing this up, and this will hopefully be available for everybody soon. And just to give an idea, so these are the different data sets going across at the top, and then the different MZ differences. And these are across different analytical instruments, so thermo, waters, ABCIX, etc. in there, different sample types, etc., different helic versus reverse phase versus lipidomics assays, etc. in there. And just to give an idea, just from a favourite metabolite, tryptophan, you can see, in-source fragments, you can see the protonated ion and sodiated ion, a disodiated ion, but only carrying one charge, for example, within there, and you can see the homogeneous dimers. We can also see heterogeneous dimers as well within here, which have been reported frequently. And there are many different metal adducts that you can see, not just the common sodium, ammonium, potassium, but you see magnesium, calcium, iron. All of these are in biological samples, so typically you have the opportunity for those to be formed. Okay, so that was thinking about MS1 data. So let's think about MS2 data and something I call retention time mapping. It's not retention time prediction or in silico prediction, it's retention time mapping, as you'll understand in a minute. And as you can see, I've got a lot of PhD students who are close to Viva. Annie's got her Viva in January, so she was the main lead on this work. And then Judith was also involved as well. So why do we want to perform retention time mapping? Well, we're starting to get big data available, either, for example, mass spectral libraries, or data repositories. We're getting a lot of data, a lot of different studies deposited into different data repositories, for example, metabolites, metabolomics workbench, etc. Within there. Most annotation identification work is using the MS1 and MSMS data, but we should remember that retention time data, which is often forgotten, is actually highly complementary based on the mass spectrometry data. It's based on different physical chemical properties of the metabolite. And so using retention time data is a really good tool to be able to improve your confidence of your metabolite annotations or identifications within there. Now, the availability of retention time libraries currently is limited, okay, especially if you compare that to MSMS libraries. And because different laboratories apply different UHPLC assays, then the retention times for the same metabolite can differ across different laboratories, different assays, etc. within there. And so they're not generally comparable. Now we know, because we've done some work before, and I'll show that in a minute, that can actually start to compare or calibrate between different laboratories, different assays within there. And so some work that we've done many, many years ago, again, when I didn't have a grey beard, was looking at what we called retention time mapping between two data sets that were collected on two different instruments applying the same assay in the same lab. Okay, so basically we're looking at combining, this was looking at combining, for example, two different data sets on the aqueous reverse phase within there. And we can see, and you can go and have a look at that. We've done this more recently, looking at using a Vanquish UHPLC system with acute thermochemie plus system, to do an older method and a newer method. And again, we can see really good mapping between method 1 and method 2 on here. So we can calibrate and transfer retention times from one method to the other in our lab, into a laboratory within there, with a relatively good relative mean square error within there of 9 seconds. Okay. So this is retention time mapping. It's not in silico retention time prediction, I want to say again as well. And so we've looked to see whether we can do this with different libraries. So thinking about a library collected in one laboratory, a library collected in the other, can we combine those libraries together to increase the number of retention times that we have? So we've done that with two different libraries, one for my lab, and then the Fine Hillock lab, which is publicly available within there. Used Hillock columns from different manufacturers within there, similar mobile phases, I will say. And as you can see, there is a general trend, general calibration within there, but it's not perfect. And there are reasons why these are outliers within here. Okay, and that's based on the physical chemical properties and whether they've got one or two hydroxyl groups within there. This is what we're thinking at the moment, though we're still looking at this. So it looks like we can calibrate between these two libraries, and therefore we can increase the size of each of these libraries. Okay, so we're reusing data in a new way, and which can be used currently within there. The root mean square error is higher, but it's still something that we can use to say if something has a retention time of say 180 seconds, but in our retention time library it's 400 seconds, and we know it's not that, so we can remove that from the potential list of metabolites within there. So watch this space, there'll be more work being performed on this. And then thirdly, the detectable metabolome, and this has mainly been performed by Manuel Lotz, who was a master's student in my lab over the summer. This is work that's just been completed or completed to the level it has. And Will Nash again was involved in this. And I'm thinking about the detectable metabolome versus the predicted or the possible metabolome within here. So we have a lot of publicly available data becoming available, and we have two grants with Claire O'Donovan and Tim Ebbles, and Peter Dorstein is involved in one of these, which is just about to start. We're calling these the Mariana grants because for anybody that performs untargeted metabolomics, you know how difficult it is, and you feel like you're falling into deeper problems as you go deeper into the data within there. So it's a bit like falling into the Mariana Trench within there, or trench within there. So we know early on in metabolites, metabolomics workbench, etc., that a lot of the metabolites that reported weren't annotated. And really that's because either the pure chemical standards aren't available to purchase, so you can't develop the retention time in MSMS libraries within there. So we have a very large search space at the start of what is the possible metabolome, but actually we know that a lot of those things we don't detect. So can we improve our metabolite annotations identifications by sequencing the metabolomes or sequencing the detectable metabolomes within there? And so here's just a very, very nice example. So in red here, we've got the potential complete metabolome, okay? And then in blue here, we've got the detectable metabolomes. This is what we frequently detect in a specific biological sample, for example, human blood or human tissues, human liver, for example, within here. And the size of the red and of the blue is both dependent on the sample and on the assay that you apply for this within here. And I'm not saying that this blue circle here is this large. It could be smaller, it could be larger, depending on the sample and the assay that you're applying within here. So what Manuel did, he took about 120 data sets from the metabolomics workbench, and basically integrated all the metabolite annotations in those data sets together, and then listed those which were reported most frequently. So he applied not smiles on these, he applied inch keys on these, and then used these to filter through. So these are unique metabolites. So these are sort of the top 17, 20 metabolites that were reported, with a number of different studies that they were reported in, out of the 120 within there. So you can get an idea about types of metabolites which are routinely being detected. And all of these were untargeted metabolite annotations within there. So because they're annotations, we have to be careful in the confidence within those, but if you're getting them reported multiple times, then your confidence will increase as you go through there. And so we can start to use these, see which metabolites are more routinely detected on which types of assays within there, and therefore to provide more confidence within these. Now, a lot of these will be present as chemical standards, so not all of them are, as you go further down the list. So this is going to help us potentially develop semi-targeted assays. And just to give an idea of the diversity, and this quite surprised me actually within here, but here we're looking at human blood in blue versus human tissues and cells in green. So we've got 734 unique metabolites just detected in blood products, so serum, plasma, dried blood spots within here that were unique to blood. And there was 248 which were unique to tissues and cells. And then there was an overlap of 539 between those. But there's a number of metabolites which are not detected, which are detected in tissues and cells, but not in blood products. And there were a higher number of metabolites which are detected in blood products which aren't detected in tissues and cells. So we're thinking here about the exposome, etc. within there as well. So that has to be, you have to think about that and be careful about that when you're developing these types of assays as well, and thinking about interpreting the data. So just to thank everybody in the group, and just to summarize that metabolite annotation or identification can be a difficult task, or there can be hurdles in untargeted metabolomics. This is why we're moving more towards semi-targeted assays in metabolomics studies. And I've shown you some ways to help you think about the complexity of the data, how to reuse retention time libraries, and how to think about defining what's most commonly detected and using that to increase your confidence in metabolite annotations or identifications. And thank you again for the invitation, and thank you for listening.
Thank you so much, Rick. Beautiful talk. You're getting questions. You want to ask the question? Unmute yourself and. No, we can't hear. Okay, I'll read the question. What is your estimate of the size of the whole human metabolome?
Really good question. Do we really know? If you think about everything that you could be taking, we have a really good idea about endogenous metabolism. We have sort of an idea about what comes in from food, etc. But when you go to the exposome as well, then it starts to expand even more and more. So, you know, the human metabolome database is 114,000 metabolites at the moment. Some of those are predicted, some of them have been detected. So we are talking about hundreds of thousands of potential metabolites or chemicals. Now again, but it comes down to that point that most of those we never detect, at least with untargeted studies. As you go down in concentration of those chemicals, and you develop targeted assays for those, you'll start to detect some of those lower concentration metabolites.
Any questions from the audience? While we are getting Rick, I want to ask, you mentioned the retention time shift. It with the column. So if we have, that mean we have to get the column from the same batch if we doing a large analysis, large number of samples? So,
so retention time shift, I meant really across different assays within there rather than within an assay. The assays that we've developed, and you see this in other assays that have been published, pretty much the UPLC, UHPLC columns we buy now are relatively stable. And in the Phenome Centre Birmingham, at University of Birmingham, where I used to work, we looked at this over 2 years, and we're seeing retention time drifts using the same column, but not necessarily the same batches of that column of 2 or 3 seconds. So it wasn't really huge. As long as you set up and you equilibrate your system really well, then you don't see a large drift in retention time.
Okay, that's good good to know that, because it's very important. Any questions coming for Rick?
So either too early or too late.
You explain very well. Everything is there. Okay. Probably we might get questions later, because we can stay around. Thank you so much, Rick. And so I'm thanking all the speakers today. Now it's time to give an award for the lightning talks. Exciting this year. We got today's sponsor is Metabolites. So I will share my screen once again. So the Metabolites is an open access journal by MDPI. So the impact factor is 4.932, index in PubMed. And Professor Peter Mikkel, this morning our keynote speaker, is the editor-in-chief. So what we are planning is, so this is, go to the next slide. So we are planning to have a special issue. If you would like, if you are like to submit an abstract or your manuscript next year by 31st of March, please contact us. So we are happy to prepare a special issue based on this virtual podium 2021. So without further delay, I want to give an exciting information. So today, based on the presentation, award for the best lightning talk sponsored by Metabolite is the Daisy Saho.
Oh, thank you so much.
I wish we can give awards to all the 4 speakers. That's so good, but unfortunately, based on the committee, so we get only Daisy. But there are other awards waiting. So we'll announce it end of the session for the day. Yeah, and thank you so much for all the speakers today, and Erin and Rick and all the 4 lightning speakers. A wonderful session and lots of information. But if you want to stay around and share the information or meeting, I will leave the Zoom link open for a while. Yeah, thanks again for all the participants and joining this difficult time for some of participants in the world. Yeah, thank you. Thanks, Rick.
Thanks. Hopefully I'll see you soon.
See you soon. Thank you so much.
Thanks. Bye.
Bye.
Lucy, how is it going?
Yeah, because Belle couldn't give the she couldn't do the chairing, so I had to jump and do it. I hope everything went okay. I'll stop recording.